Okay, I'm back with actual melted cheesecake now. Or it's not melted, but it's not frozen like it was before. So I get to eat that. While we discuss this. So good. And I did get a chance to look at my last page of notes. And I kind of understand it. Not too much, but I think I have a basic understanding, like to the point where I can take the quiz, but not like definitely like not test level. Mm, but I'll go over it because I need to. And I'm about to take this test or quiz. I was fixing on taking it earlier, but then I laid on my bed and I listened to music and I passed out. So and this is where it's at right now. I'm gonna have one more piece of cheesecake, or like I'm gonna finish this piece. It's just one, but I'm gonna finish it, and then we're gonna move on to more terms about evolution and phylogeny. I don't know what type of teacher eats during their class, but that's me right here. I don't know if they allow teachers to eat during class, but I'm not a teacher and I'm eating, so and it's not even a class. But if I were to be a teacher, I would love to be able to eat. But yeah, it is kind of hard to teach while you're eating. Okay, 3-minute mark. I think that's good enough to start working on the notes. Okay. So going back into the notes that I wrote for biology. So last lecture we talked about the phylogenetic tree, dendrogram, branching picture, whatever you want to call it. And I mentioned that the little intersection branching points each represent a common ancestor in the dendrogram, and this is just a continuation of the notes that I wrote for that, but I forgot to mention that the branch links can indicate evolutionary rates and time. So I already mentioned. You'll hear that. I have to check if that's my car. Let me see. That sounds like my car. Sounds like my BMW. Okay, so it stopped, so I don't know. It sounds like my car though. We'll check that in a bit. This shouldn't take too long. So, anyways, I already mentioned that each part of the branch could represent any of the ancestors, and but I didn't get to mention that the branch links can indicate evolutionary rates and time. So, how long the rates and how long the how long it took for the animal to evolve and stuff like that. Okay, so we have, I think, 5 types of traits to go over. So the first one is apomorphy or antapomorphy. Antapomorphy literally means derived. So this is a specialized trait unique to one group or species. So example of this is erect posture in humans and mammary glands in mammals. The second term I would like to go over is plesiomorphy. That's a trait found in common ancestors passed on to some but not all groups, or like some if not all groups. Let me see. We can find an example of that. I have drawn a graph, but I don't know if it's like okay. I think I know what I'm. I would like to know the difference between plesiomorphy and symplesiomorphy. But what would be what? Here. I get it. I get it now. Okay, so it's wait. I have to read that again. Share, please. Okay. All right. So I got it. I got it. Okay. So plesiomorphy is just a shared trait found from like a common ancestor and the derived species. Okay. Oh, excuse me. So example is hair and backbone in humans, and then we have synapomorphy, which is should be which is like shared and derived. So let me see if I can. I just. This is so confusing. Sorry about that. Okay, I guess synapomorphy would be like apomorphy, but with the ancestor. So like, so I mentioned that apomorphy has a trait, but it's unique to the species, so it's derived or whatever. But synapomorphy would be that same thing, but with the ancestor. If that makes sense. And then the next one we have is symplesiomorphy. So I just mentioned plesiomorphy is a trait found passed on from an ancestor, but this symplesiomorphy is just basically like when two groups have the same, like there's two branches and they have like similar traits. So it would just be like multiple plesiomorphies. So I guess to memorize or to remember is like symplesiomorphy is multiple plesiomorphies, and plesiomorphy and symplesiomorphy are ancestral, whereas apomorphy and synapomorphy are derived, is what I'm trying to say. And then we have homoplasy, which is two same characteristics that show up in random places on a phylogenetic tree. So it's like random, like there's no really like, it's not really like a derivation or an ancestral thing. So I made this chart. It looks kind of funky, but we're gonna work our way through it. So it's very hard to see. Oh, this is not clear at all. Not clear at all. How about I'll just try my best. Okay. So, okay, phylogenetic tree, right? This is A, this is A, this is A, B, C, and D. So we first went with apomorphy, which would be right here. D right here. It's like just that. Just this right here. Okay. And then we moved on to plesiomorphy, which I'd say this is a plesiomorphy, if nothing else. Like A right here, that stems from A. And then symplesiomorphy is like right here. Because see this branch, it comes right here. And this is a different branch completely. And it still kind of went that way. So this would be two of the same plesiomorphies because they came from the ancestors. My voice is dying right now. So yeah, that's plesiomorphy and symplesiomorphy. And then that's apomorphy, and this is synapomorphy. Why? Because oh yeah, okay, so like it has the ancestor right here. So synapomorphy. And apomorphy is just unique to the group. Synapomorphy has the derived characteristics from the derived species, but also the ancestor. And then homoplasy, I think. It shows up randomly. It's like, so you see this one, this B stemmed from this branch of A. This is a completely different branch, but it still ended up with B, the B trait. So that's homoplasy because it's just like it's like random spots on the phylogenetic tree. I hope I could be able to see that in the future when I look back at this video, but it's probably not going to be visible. So that's really, really, really annoying. But that's just how the way technology works. Okay, moving on, we have two types of DNAs that we can use to detect these. To study these changes and detect these changes. So we have a DNA that codes for RNA. So we have the ribosomal RNA. That DNA or that RNA. Oh no, sorry, that DNA that codes for our RNA changes very slowly. So we can use this to detect changes over hundreds of years. So it's like pretty easy to detect, I guess, because it's like slow changing. But the DNA that is in the mitochondria, so the mtDNA, it evolves super rapidly. So we could use this to explore more like recent evolutionary events. So like we can use this to study like, okay, what's happening right now? So I guess like we can use the RNA DNA to look at the past, and then the mtDNA to look at what is in the present and possibly what's in the future. I have a term that says gene families, but I didn't get to write down the definition before my professor switched the slide, so I guess we're just not going to go over gene families, but just search up what it means. This shouldn't be that hard. Okay, then we have two types of genes. We have orthologous genes and orthologous and paralogous genes. The first one, orthologous, is the exact same gene in different species. Okay, I think I get what I mean. Okay, so it's the exact same like gene in different species. Yeah. So example is cytochrome C oxidase in humans and dogs. So like humans have it, dogs have it. And then yeah, the same gene, sorry. And then we have paralogous genes, which is multiple copies of genes diverge from one another within a species. So example is olfactory receptor genes in vertebrates. I mean like and then the example says 380 copies in humans and 1,200 in mice, but like humans and mice aren't the same species, so I don't really know what. I mean, I get the idea, but it's not the same species. But I guess the idea is that they evolve like for some species to have different types of genes. Like it's the same one, but it's different. I don't know if that's a good. I don't know if I understand that, but we're gonna move on. Okay, so we have this thing called a molecular clock, and this is a method to estimate time required for a given amount of evolutionary change based on observation that some regions of genomes evolve at constant rates. So this is just like kind of like a way to like estimate evolutionary change, like just to kind of look at evolution and more like measuring, just kind of like put a number or put a label on it. But obviously and quite evidently with like multiple methods of like kind of like putting things into a box, especially in science, like using the word always or never, it's often there's often problems with it, like in science or in anything in life really, but especially in science, things aren't always what they seem to be. Like, you know, oil isn't like entirely insoluble in water. Like there's some oil that does dissolve in water, but there's not like a lot. So we call it like insoluble, but it's not actually insoluble. So that's something to think about. So like definitely in science, you can't really just put everything into one category and just call it a, because a lot of things are they're this, but then they're also that, you know? So what are the two main problems with a molecular clock? Well, the first problem is genetic changes may occur at irregular rates instead of a constant rate. Like the molecular clock is based off of the assumption that the evolution will occur at a constant rate. And the thing is, evolution doesn't always occur at a constant rate. In fact, I don't think it ever really occurs at a constant rate. It's just a way for us to kind of like estimate and like kind of, you know, use averages and stuff to call it average in a constant rate. But I don't think evolution ever, it doesn't really work that way. It's nature. Second one is molecular dating techniques may not reflect paleontological dating techniques. So yeah, like what is in theory like the genes that we change, like that change doesn't might not really like reflect what happens in real life. Like what we study in the lab doesn't really, like it might not reflect what happens in real life. So what happens in our blood, it might not like reflect in our our outside. So for example, like you could have a recessive gene for a disease, but you not actually have that gene. Or that gene doesn't actually show up. You don't have that disease, but you still have the gene inside of you. So you could still inherit that from your parents, but it's just not going to show up until maybe like a later time or something like that. And just to kind of go back to some of the taxa that we went over. I have some things to go over. Okay, so we're going to go over some domains and kingdoms, right? So we have, for domains, we have eukaryotes and bacteria and archaea. So bacteria and archaea are prokaryotes, but we're not going to call it prokaryotes. It's just separate into three different domains. And the kingdoms, so we have the eukaryotes. The kingdoms are animals, protists, fungi, and plants. Plants. Yeah. And then I'm sure there are there's like archaebacteria, and then there's like some other kingdoms, but I didn't write it down, and we're probably gonna get into it at a later time. So yeah, I think that is it for now. I'm gonna take the quiz, do a pack back discussion, and I'll be done for the day for Sunday. Really excited, and I'm glad I got to eat my cheesecake. This was half the amount of time it took for me to film the other one. So it was 2 pages? 2 pages, 40 minutes. Okay, so I'm just gonna assume that each page that of notes that I write, for me to properly understand it and to retain the information and teach myself the topics efficiently, I'd probably have to spend like about 20 minutes per page, like talking it out. But I also get distracted and I get carried away, so yeah. But I will go get back to business, and hopefully I will get an A. I'm just going to automatically assume, like, is it safe to automatically assume I got like an A or 100 on every single one of my videos or like my assignments that I write or that I create lectures for, unless I write down that I didn't get the 100? Because I want to like record the times I don't get 100 so I can like know what I got wrong and know what I need to review in the future. But yeah, I'm going to go do my test and then probably walk around a bit. I didn't get any exercise today, and then I'm gonna go sleep. All right, bye.